Welcome to the third lecture of the week. In this lecture, we will discuss chromosomal or abnormalities or changes in number or size or shifting within the chromosomes. And then we will talk about various conditions that arise due to these alterations in the chromosomes. So I hope you enjoy the lecture and let's begin. So the objectives for this lecture would be to know the different types of chromosome alterations. You should then be able to describe polyploidy, aneuploidy, and non-disjunction. And then finally, we're going to talk about some chromosomal alterations and the conditions that arise from them. So you should be able to detail and identify these conditions related to the chromosomal alterations. So what do we mean when we're saying chromosomal abnormalities? So chromosomal abnormalities are when you are altering a chromosome by losing, adding extra, or you have an irregular pattern of DNA within the chromosome. Often there is either a shift or a loss or a deletion, which we'll talk about, and you're altering the chromosomal number. And as a reminder, we look at chromosomes and their layout within an individual using the karyotype as listed over here on the right, where you can look at all the 23 pairs of chromosomes. Remember the 22 autosomal, and also the sex chromosomes, X and Y, as you can see here in this male individual. This is a normal, unaffected, wild-type male karyotype, where you have even homologous pairs, and it looks very good. But we'll see how there can be changes in this pattern and what conditions that arise in the remainder slides of this lecture. However, in general, chromosomal abnormalities often lead to mental retardation, and actually a vast majority of them with these chromosomal abnormalities lead to a miscarriage. The developing embryo just doesn't make it to a stage where it becomes a point where it continue growth, and so they are often miscarried early in the developmental period. As you see, almost 50% of all first semester miscarriages are associated with abnormalities, and it is relatively common though to be born with a chromosomal abnormality, in that 1 in 150 live births have chromosomal abnormalities. And we'll talk about some of the more common ones like Down syndrome in the later portions of this lecture. So I just wanted to have a few basic introduction to the chromosomal alterations, and some of them are kind of straightforward, but I'd just like to walk through. So a deletion in the chromosome would be a loss of genetic material. So you can see here, these are model chromosomes. We'll use a similar pattern in discussing all these alterations. And so in this instance, we have deleted this end or portion of the chromosome. And so you can see that fragment has been lost from this chromosome, leading to a deletion or a loss of genetic material in this chromosome. You can also have a duplication. And so you would have a section of chromosome becomes copied and then duplicated in another chromosome. So you'd have an extra copy of the genetic material, which obviously could lead to higher levels of expression of certain genes, leading to all sorts of downstream phenotypic changes, potentially diseases. Another example of a chromosomal alteration would be translocations, and that would be when you have an exchange of pieces of DNA between non-homologous chromosomes. So remember, we talked about the crossing over that occurs in independent assortment of DNA that happens between homologous chromosomes. However, in this translocation, we're having an exchange happen in non-homologous. So what could happen here is, let's say you had a balanced unpaired chromosomes. You could have reciprocal crossing over or exchange, where you'd have this front portion of the chromosome exchanging with the bottom half of the other chromosome. So you'd have a swapping of the two in a balanced manner. So it's kind of a 1-for-1 or give and take between the two chromosomes, however non-homologous they may be. You could also have acentric fusion, in that you end up with an uneven exchange, in that you end up with a rather large chromosome due to the pairing of these two and the crossing over, and you end up losing a large or a smaller section right here, ending up with a large chromosome. So this would be another example of a type of uneven translocation that could happen. Additionally, you could have an inversion, where you have an upside down reinsertion of a selection of a chromosome, in that you're essentially looking at a chromosome that is swapping locus on that. You have regions of DNA swapping loci within the chromosome. So they're swapping positions. So you could see here in this paracentric example, you could see the blue, the darker maroon swapping with the light blue in the resulting paracentric swap. You could have pericentric swap where they're rotating around the center of the chromosome, and you can see them swapping here, leading to a chromosomal exchange, which could also lead to alterations of gene expression because you could be unlinking like a specific promoter from its regulated gene, which could cause changes in gene expression. All these have that similar risk in that you could shift a gene away from its regulatory sequence, or you could end up with swapping and moving genes around, disrupting them within the middle of the coding region of a gene that could lead to irregular proteins being expressed and potentially diseases due to that. So the previous chromosomal alterations we discussed involved movement between chromosomes or also deleting or adding or moving pieces of chromosomes around. Now we're going to talk about what happens when you have extra copies of chromosomes. And we call that polyploidy. And so that means when you're having extra copies. And so euploid is a cell that has a multiple of 23. And we have some subcategories of euploid. So a triploid or triploidy is where you have 3 times the normal amount of chromosomes. You would end up with 69 chromosomes. This can happen if you have an instance where called dispermy, in that the egg is fertilized by 2 sperm cells at the same time. So you'd have the 23 chromosomes from the one sperm, 23 chromosomes from the other sperm, and then you combine with the 23 chromosomes from the egg, you end up with the 69 total chromosomes. Another way this could happen is if a polar body somehow fused with a fertilized egg or an oocyte, ending up with the fertilized egg. So the fertilized sperm, or the sperm having 23, the fertilized egg having 23, and then the polar body somehow adding on to that, adding another 23, ending up with the 69 chromosomes as we described. You also have tetraploidy, which would be 4 times the normal chromosomes, or 92 chromosomes. And this happens when you have the fusion of diploid zygotes that have been, zygotes are fertilized embryos on their way to becoming developed. Fusion of these together at this stage would lead to a tetraploid embryo, which would have these 92 total chromosomes. These are obviously not viable and have usually have issues developing the extra embryonic tissue, so they don't develop like the placenta, and so they miscarriage very early in pregnancy. Euploidy was when we had multiples of chromosomes. Aneuploidy is we have an abnormal number of chromosomes in that it's not a multiple of 23. So a few examples of this would be trisomy, where you have 3 copies of one chromosome. So we'll talk about trisomy 21 in a few slides, which is classically known as Down syndrome. You could also have partial trisomy, where a small portion of the chromosome is in excess. So you'd have a fragment of the chromosome be replicated added to that. You also have instances where there's mosaicism, in that only a small percentage of the cells or a portion of the cells in the developing organism has the trisomy. You'd have that triplication, but they don't all have that. Remember the mosaic we talked about in the last episode, or last lecture, where some eggs or some cells have the gene, but some don't, depending on if it's turning on or turn off with that X inactivation. In this case, some cells would have the extra chromosome and some would not. Monosomy is where you only have 1 copy of the chromosome. This is usually lethal. You'll usually need both alleles to allow for normal expression. Also, it could lead to you being exposed to a host of recessive genes that could lead to issues in the overall development and viability of that developing organism. And finally, the main cause of a lot of these trisomies or monosomies is a process called non-disjunction, which we will discuss in the next slide. So what is non-disjunction? Non-disjunction is the failure of chromosomes to properly separate during meiosis, leading to an uneven number of chromosomes and often a trisomy-like state. So if we look at our normal meiotic division, we have in meiosis 1, we separate the homologous chromosomes. Meiosis 2, we separate the sister chromatids, leading to an even distribution of all the cells being even ratio of chromosomes. However, we can have non-disjunction either in meiosis 1 or meiosis 2. So if we have non-disjunction in meiosis 1, you can see here, the sister, or the homologous chromosomes, fail to separate properly. So you have an uneven separation in that this pair ends up moving all to the daughter cell on the left. This essentially subtracts chromosomes from the daughter cells on the right side in meiosis 2, leading to an excess, or the trisomy, or extra copies of the chromosomes in the gametes. And so these are +1, an extra chromosome, and these are missing a chromosome. Non-disjunction in meiosis 2, you have the even separation of the homologous chromosomes in meiosis 1, but the sister chromatids in meiosis 2 fail to separate properly. And you can see here in this state, you end up with one cell having an unequal number of chromosomes or an extra chromosome, while this gamete cell would have one less chromosome than normal, and then you would have two normal cells because this side did separate properly in meiosis 1. So non-disjunction is this uneven separation of chromosomes that leads to imbalance in the number of chromosomes at the gamete level, which would be either the sperm or the egg. So now I wanted to go into a few examples of what conditions are associated with these trisomies caused by the non-disjunction we previously discussed. And a very common, in fact, the most common abnormality would be trisomy 21, which is commonly known as Down syndrome. As you can see here in our colorized karyotype, we can see you have an extra copy of chromosome 21 shown here in this male individual. There's a lot of literature and research that has determined that there is an increased risk of trisomy 21 with maternal age. That is, as the mother gets older, the chance of having offspring with Down syndrome increases. In that, if you look at the sheer numbers, if the mother is less than 25, there's a 1 in 2,000 chance of this occurring. If the mother is between 30 and 34, there's a 1 in 900 chance. And if the mother is older than 44, there is a 1 in 46 chance. So you can see the correlation between the mother's age as increasing along with the risk of trisomy 21 occurring. Now, some of the symptoms of trisomy 21 would be general mental delay or mental retardation, in that these individuals have a lower IQ in the range of 25 to 50. They often have a protruding tongue and low set ears, as you can kind of see demonstrated on this individual. They have also epicanthal folds, which are folds on the inner lining of the eye, kind of starting to fold over a little bit from the medial plane towards the lateral plane of folds of the eye. In general, these individuals have poor muscle tone and shorter stature. Unfortunately, they are affected by congenital heart deformalities, so they have a high risk of heart disease and heart complications. They also have increased susceptibility to infection and leukemia, various immune responses. And unfortunately, these individuals by the age of 40 really do start to develop Alzheimer's disease, which is a very crippling neurological disorder that leaves them losing a lot of their memory and some of the skills that they are already somewhat delayed on due to the mental retardation associated with trisomy 21. Another trisomy example would be trisomy 18. This is the second most common trisomy, affecting 1 in 6,000 live births. Similar to trisomy 21, it does have an increased risk as the mother ages as well. So as the mother gets older, you have increased risk in general for these trisomies, and trisomy 18 is included. The phenotype for these individuals would be growth deficient. They have a prominent occiput, which is a prominent back part of the skull, as you can see here. They have a lower ear, similar to in trisomy 21, and they also have a smaller mouth. Congenital heart defects are also common in this trisomy, and there is an associated mental retardation. Unfortunately for these individuals, trisomy 18 has a much higher mortality rate, in that only 5% of these babies born with it survive to the age of 12 months. So it is a very damaging and unfortunately very disastrous chromosomal abnormality to be afflicted with. The third trisomy I wanted to bring up would be trisomy 13. This is a little less common in that it's only in 1 in 10,000 births. Similar to all the other trisomies, there is an increased risk with maternal age. In these individuals, they have smaller and abnormally shaped eyes. They have a lot of eye malformations associated with them. Cleft lip and palate, as you can see on this diagram example, is common. The heads appear to be smaller. There is also a mental retardation, which these individuals, post-axial polydactyly, like we mentioned before and we talked about in our autosomal pattern of inheritance, you can see extra digits on the pinky side with that post-axial polydactyly. And there are also cardiac defects. Similar to the previous trisomy 20 or trisomy 13 has a very low survival rate for babies born with this, in that only 5% survive to 12 months. So trisomy 21 being the exception, many of these trisomies are very lethal, at a very early age. So showing the severity of these chromosomal abnormalities. Another example of a trisomy would be Klinefelter syndrome. So these individuals, you can see an example here, are born with a sex chromosome abnormality where they have extra copies of the X chromosome. So they could either be XXY, XXXY, or XXXY. It's somewhat common that 1 in 850 live births are exposed to Klinefelter syndrome. It is often not observed until puberty, when the male secondary sex traits start to develop with puberty, right? Or when the males start fuller testosterone development, is inhibited due to the extra copies of the X chromosome. So these individuals have smaller testicles that lead to decreased testosterone, so you're not developing the testosterone that you'd expect in boys in puberty. So they're not getting the secondary sex characteristics development, increased muscle mass, voice modulation changing. So they often end up with a feminine hair distribution. The voice does not change. You maintain a more feminine, higher pitched voice. And it is very common for breast development due to the extra X chromosomes is inhibiting the amount of testosterone. So the estrogen, normal estrogen levels appear to allow for some amount of breast development. These individuals are usually infertile, or in that they are not being able to produce the, probably due to the low testosterone, they are not be able to produce sperm to a level to allow for fertility. And they also have somewhat of impaired intelligence due to this condition. Turner syndrome is our next example, and that is affecting 1 in 3,000 females. So this is our one example of a monosomic condition in which there is only one X chromosome. So these individuals do not have the second X, or they do not have a Y chromosome. So they will have a female phenotype. They only have 45 chromosomes, and there would be that one X chromosome. They don't have the extra X or the Y. In this instance, it's not the maternal age, but the paternal age that increased has an increased risk. So it's usually the father can't pass on the X or the Y chromosome due to something being lost in sperm development. The symptoms associated with this would be shorter stature, webbed neck or widely spaced nipples. You can see this neck of this individual seems to be wider than normal. These individuals are sterile in that the ovarian development has halted early, and so it's not fully producing it's not supportive of oocyte development. Similar to some of the trisomies, there are congenital heart defects affecting these individuals, and as very common in all of these chromosomal abnormalities, mental impairment does seem to be a symptom. And so this Turner syndrome has that mild mental impairment as well. So that is the end of this third lecture. So you should be able to know the different types of chromosomal alterations. You should be able to describe polyploidy, aneuploidy, and non-disjunction, and really be able to explain how non-disjunction leads to those trisomies or chromosomal uneven balance, especially trisomy and maybe the monosomy as well, associated with non-disjunction. And then you should be able to discuss and identify the conditions we talked about at the end of the lecture, the different trisomies and Turner and Klinefelter syndrome, and be able to identify what those chromosomal alterations are that are associated with those diseases. I hope you've enjoyed this week. I really enjoyed discussing it and digging into it a little bit more than I have in the past. And so I look forward to reading your discussions, and I wish you all blessings on your studies.